All right, so we're going to go ahead and move on to chapter 3. Chapters 1 and 2 really were just review stuff. And while chapter 3 is going to have a lot of familiar information, am going to start moving a little slower, and I'm going to spend 3 days on chapter 3 because this is where we really start getting into the meat of the course. And so we're going to talk about the structure and function of large biological molecules. And so when I'm done with this lecture, I need you to know, the lecture after 3 days. Today, I'm going to go over dehydration, synthesis, and hydrolysis. And then I'm going to skip down here to the 4 structural levels of proteins, and then protein shape and protein denaturation. So today I'm going to discuss dehydration, synthesis, and hydrolysis. And then I'm going to discuss proteins in detail. And so here are some vocab words that you'll need to know: monomers, polymers, and macromolecules. So we have 4 macromolecules: proteins, nucleic acids, lipids, and carbohydrates. And all of those macromolecules are made up of monomers. And so monomers are considered like the little building blocks that are bonded together to make polymers, and then polymers are bonded together to make macromolecules. So the analogy I like to use is if you're putting Legos together, each individual Lego is called a monomer. If you put a chain of Legos together, that would be a polymer. And then if you put enough chains of Legos together to make an actual functioning structure, like a castle, that would be like your macromolecule. And so monomers are small and organic, and they're the building blocks. And they are connected with what's called condensation reactions or dehydration synthesis. And I'm going to go over that word in just a minute and what that actually looks like. Polymers are going to be long chains of monomers with many identical or similar blocks linked by covalent bonds. And then your macromolecules, and I told you what those 4 macromolecules were, are 2 or more polymers bonded together. So you could start small, larger, and then largest. An example would be amino acids are the monomers that are bonded together to make peptides. Peptides are then bonded together to make polypeptides. So peptide and polypeptide really are interchangeable words. And that could have actually been just one word right there. But peptide and polypeptide are your polymers, and then protein is going to be your macromolecule. 2 reactions that you need to know are called dehydration synthesis and hydrolysis. Dehydration synthesis is also known as a condensation reaction. It is the process used to make polymers, to link monomers together to make polymers. And so A plus B bond together to make AB, and water is actually like spit out as a byproduct. All right? So on this last slide, when amino acids are bonded together to make a polypeptide, it is using dehydration synthesis to do that. And water is actually a byproduct that gets taken out. So why water? A is going to have a hydrogen hanging off. B is going to have a hydroxyl group hanging off. So that's an H and an OH. Take the H off of A and the OH off of B, you get water, and then A and B bond together. That's kind of the basis of that. And I'm going to show you a picture in just a minute to kind of help you understand that. Hydrolysis is the breakdown of polymers. It's the opposite. Water is used to break a bond. So a hydrogen gets hooked onto A hydroxyl gets hooked onto B, and that bond breaks. So hydrolysis is the way that polymers are broken down, and it's the insertion of water into a bond that breaks it down. And so this picture right here is showing you, and hopefully I didn't go too fast on that. If you need to pause, go ahead and pause to write that down. Right here is showing you a dehydration reaction. And so a dehydration reaction, the H on one monomer and the OH on another monomer join together, water gets spit out, and in that process a bond between these 2 are made. All right? Hydrolysis is the opposite. So we want to break this bond, water is put in, H goes to the 3, OH goes to the molecule that was labeled 4, and that broke the bond. And now you have a bond broken, obviously. So dehydration reaction is the building, hydrolysis is the breaking down. So you're going to make sure you understand that and that you know it. moving forward, because that does come up again. All right, proteins, and this is not page 54 for you, this is page 32. So if you can cross that out, this is page 32. And obviously you already saw in the description for this assignment that the notes started on page 31. We are now on page 32, and we're going to talk about proteins today. So proteins come from the Greek word proteos, which means first or primary. Proteins I consider to be the most important macromolecule, and it's the one we will talk about all the time. So just go ahead and learn it now, because it is part of everything that we talk about in this class, because your traits are determined by proteins, and evolution is driven by proteins also, you'll see. And so proteose means first or primary. Proteins make up 50% the dry weight of cells, so that's a lot. How you're going to recognize a protein is that it consists of the atoms CHONS, chons. Now, just one amino acid might not have a sulfur, but a protein is usually made up of at least 400 amino acids. And so your amino acid cysteine, which does have sulfur, is going to pop up somewhere in there. So every protein is going to have a sulfur at some point. And this just shows you the model that we use when we show what a protein looks like. You'll see spirals, you'll see pleats usually. And so if you see these spirals, that's a protein. Or if you see a purple glob, that's also a protein. So those are two models, are these spirals in pleats, and then a purple glob. So anytime you see a purple glob, that's definitely a protein. So this is showing you the myoglobin protein, and this is what carries oxygen to muscle cells. All right, protein functions. You don't need to write anything down because that is already listed beneath where it says protein functions. I left a big space, but you don't need to write this down because it's already there for you. Okay, so I'm going to skip over this because it's already there for you. And I'm going to go through each one of these little boxes. So proteins are the most versatile of all the macromolecules. They have the most jobs. They can function as enzymes. Enzymes are going to catalyze reactions, and they're going to speed up the rate of reactions by lowering activation energy. You should have learned that in chemistry or cell biology. We will have a chapter over enzymes, so we'll definitely go over this again. But basically, an enzyme is a catalyst for a reaction to occur, and it speeds up the rate of the reaction, and you're going to recognize that you have an enzyme because it's going to end in the letters A-S-E, ACE. So an example of an enzyme would be lactase. Lactase is going to take lactose, which is a disaccharide sugar, and it's going to break the bonds when you're digesting food like milk or something like that. All right. All right, proteins can also function as defense molecules like antibodies. Antibodies are proteins that surround a pathogen, rendering it inactive. And so when a person has had COVID, or when a person is getting the vaccine for COVID, what they're going to test for a month and 2 months and 3 months later is, does the person still have the antibodies? Does the person still have the proteins that respond to and remember the virus? All right, proteins are going to act as storage. Casein and ovalbumin are storage proteins found in eggs. And so they're the storage of amino acids. Casein is the protein in milk. It's a source of amino acids for baby mammals. And plants have storage proteins in their seeds also. Ovalbumin is a protein in egg white, and it's used as an amino acid source for developing embryos. So I need you to know that proteins can function as storage. You need to know that proteins can function for transport, and the major example for this is hemoglobin. You also have proteins embedded within the cell membrane called aquaporins, and lots of other proteins embedded in the cell membrane that function in transport also. So when we talk about the cell membrane, proteins functioning for things to be transported into and out of the cell is going to become very, very important. Hemoglobin is a protein that contains iron that is in your blood, and what it does is it grabs onto oxygen in the lungs and it carries it to to all parts of the body, because we all know at this point that oxygen plays a really important role in cellular respiration. Oxygen is the final electron acceptor and cellular respiration. We are going to have a chapter over cellular respiration, so you get to see that again. All right, and then other proteins function as embedded in the membrane and function for transport across the cell membrane. All right, hormones. There are some protein hormones like insulin. Insulin is a hormone. It's a signaling molecule sent through the blood, and it's going to regulate blood sugar. Proteins can serve as receptors, and a receptor is a protein on the cell membrane that is going to capture a chemical signal, and it is going to result in a response for the cell. So an example would be your neurons. One neuron, this is one neuron right here, releases a neurotransmitter. That neurotransmitter docks onto the receptor of another cell, and it then sends a signal through that cell to do something. All right, we will have a chapter on neurons and neurotransmitters. We will also have a chapter on cell signaling, and so proteins acting as receptors. They grab onto signaling molecules when cells are trying to talk to each other, okay? And that's going to become really important. We will have a chapter later on over muscle contraction. And so contraction and motor movement, and so basically just movement. Proteins are going to function in movement. Actin and myosin are two proteins that your muscles consist of. And then the cilia and flagella, which are the little thingies that hang off the outside of cells that allow single-celled organisms to move. Flagella, bacteria have flagella, amoebas have cilia, sperm cells have flagella, so proteins play a major part in movement. And then they play a major part in structure. So keratin and collagen are two really important proteins involved in structure. Keratin is going to help with structure of your skin, hair, and nails. And then collagen is going to also function. Collagen and elastin are going to provide a framework for connective tissue. So collagen is the stuff in your skin that makes it like really elastic, and it's what breaks down as a person ages. So when people age, some people get collagen like injected into their face to try to make them look younger. It's usually obvious though when they do it, but sometimes it can look really good. So I'm not knocking it. Okay, there are 4 levels to protein structure. This is so important. Do not skip over this. Make sure you pay close attention. Like I said, proteins are the most important macromolecule, so I would ask you probably the most questions over proteins. I ask questions over all of them, but I really like to focus on proteins. The primary structure. So this is the order that proteins go in as they fold. So primary structure of proteins are just the sequence of amino acids, and they are covalently bonded together. So amino acids, and there you go, you see the first 3 letters of the amino acid that gives you the sequence of amino acids covalently bonded together, and I need you to know the covalent bond between amino acids are called peptide bonds. I also need you to know that there are 20 different amino acids, all right? And so I have given you, I'm just trying to make sure I don't go too fast. So I've given you primary structure. I've given you this picture. I need you to write down amino acid sequence. I need you to write down that there are 20 different amino acids. And then I need you to write down that peptide bonds are linked, or what link amino acids together. So if you don't have that down yet, push pause now because I'm about to move on. All right. An amino acid is going to have this structure right here. So notice you have a nitrogen, a carbon, an oxygen, and some hydrogens, okay? And then you have this thing called R. The R is a side chain that is different among the 20 amino acids, but all 20 amino acids are going to have four things. They're going to have an R group, which is a side chain, which is unique to that amino acid. And I'm going to show you an examples of that in a minute. R groups can be hydrophobic, R groups can be hydrophilic, R groups can be acids and bases. All amino acids are going to have an amino group, a carboxyl group, and then a central carbon. So when I said they have 4 things, here's what I meant. They have an R side chain. That R side chain is going to be unique to that particular amino acid. It can be hydrophobic, it can be hydrophilic, it can be an acid or a base. And I'm going to show you a list of all amino acids in a minute, and it's going to be very important for your activity this week. All amino acids, the second thing they all have is an alpha central carbon right here. The third thing that they all have is an amino group right here. And then the fourth thing they all have in common is a carboxyl group, also notated as COOH, but it looks like that. So if you have this structure, you have an amino acid. Here are the 20 amino acids. When I was in school, I had to have all of these structures memorized. You don't have to have all these structures memorized, but I need you to understand what it means for an amino acid to have a non-polar R group. So these are 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of the amino acids are hydrophobic, and they're hydrophobic because of the particular R group. And what you should notice about the hydrophobic R groups is they are all hydrocarbons. Hydrocarbons. So if there's anything that you can get into your head from today and what we did the last lecture, hydrocarbons are hydrophobic. They do not like water. They are non-polar. They don't have partial charges. They do not like water. They will go away from water and they will kind of all bunch up together. All right. You also have hydrophilic amino acids, and there are 1, 2, 3, 4, 5, 6 hydrophilic amino acids. What do these R side chains all have in common? I see a nitrogen and an oxygen, oxygen, nitrogen, oxygen, sulfur, oxygen, oxygen. They all have electronegative atom nonmetals, electronegative nonmetals somewhere on them. They are all polar. All of these R side chains are going to have partial charges. They're hydrophilic. They're polar. I would be writing this stuff down if I were you. Definitely. Polar, partial charges, and then I would definitely write non-polar up here, hydrocarbons. Then you have these guys down here. You have 2 acids and you have 3 bases. Acid, remember, gives away a hydrogen. A base is going to gain a hydrogen. And so the amino acids are going to donate a hydrogen to the base. And on this list, you can already see these acids have already lost their hydrogen and they have a negative charge. These bases have already gained a hydrogen. They're going to have a positive charge. And what you're going to see is what's called salt bridges, or ionic bonds are going to form between acids and bases, amino acids. Okay, here is how a peptide bond is formed, and this is still on page 33 to the side. A peptide bond is formed between the carboxyl of one amino acid and the amine group of another. I would write that down. The carboxyl group of one, the amine group of the other. The OH on the carboxyl and the H on the amine combine together to form water. Water leaves, boom, peptide bond between the carbon of the carboxyl and the nitrogen of the amine. You definitely will need to know that. I am asking questions on that. All right, so that is primary structure. Primary structure, the order of amino acids that are peptide bonded together. Okay, secondary structure of amino acids is how it starts gaining its 3D shape. These are hydrogen bonding between amino acids. Hydrogen bonding between amino acids. And this is going to be the hydrogen bond between a hydrogen on the amine and the oxygen on the carboxyl. These are the basic components of amino acids interacting together. These are not R chains interacting yet. These are the carboxyl and the amine groups, and I would write that down. The carboxyl and the amine groups between adjacent amino acids hydrogen bond together to form alpha helices and beta pleats. And you see those dotted lines? Those are hydrogen bonds. So if you didn't write that down, you definitely need to pause and write down the hydrogen bonding between carboxyl groups and amine groups of adjacent amino acids. And this picture shows you the same thing. All right. Tertiary structure, which is one of the most important. So what I need you to write down here is that tertiary structure is the interaction between R side chains of amino acids. Interaction between R side chains of amino acids. And then you will need to either write these down word for word or kind of fill in what's not on the picture. So you're going to need a pause because I don't want to just sit and wait for that. But this is actually on the pictures for you. Hydrophobic amino acids are going to bury together on the interior, and those are going to have van der Waals interactions. Hydrophilic amino acids are going to be on the outside of the protein, and they're going to be hydrogen bonded to each other. And that's because they're in an aqueous environment. Acids and bases are going to form salt bridges, and then cysteines are going to form covalent bonds between the sulfurs. Let me show you a picture of this. So if you want to write those down, push pause to write them. All right, here we go. Hydrogen bonds between the carboxyl groups between adjacent amino acids. Hydrogen bonds between adjacent amino acids. Hydrophobic van der Waals between the hydrophobic amino acids. Disulfide bonds, which are covalent bonds between sulfurs of two cysteines. And then ionic bonds between the acids and bases. This is what's making the protein fold. This is the tertiary structure of a protein. All that long chain of amino acids, they're going to start interacting with each other. And these are the ways that they do it. Hydrogen bonds, hydrophobic interactions, ionic bonds, and disulfide bridges. Okay. So pause right there if you need to write this down. I mean, it's all in the picture, so if you don't need to write it down, you don't have to. But go ahead and pause if you do, because I'm trying to keep this under 25 minutes. So that's going to cause folding. Quaternary structure is when you have multiple tertiary polypeptides joining together. So you have it. This is a tertiary structure of one polypeptide, a tertiary of another, tertiary of another, tertiary of another, and that's hemoglobin. Hemoglobin consists of 4 polypeptides. I would write the number 4 next to hemoglobin. That's a really important number for hemoglobin. Collagen is going to have 3, and these are kind of long chains right here. But the quaternary structure is then when it becomes a fully functioning protein. Some proteins can still function as tertiary, but most of the time they're going to be quaternary. Okay, those are the 4 levels of protein folding. And just to do a summary, primary structure are the amino acids in the sequence of amino acids. The carboxyl group and the amine group form hydrogen bonds, and that starts creating alpha helices and beta pleats. Then you have those 4 interactions between the R side chains that you wrote down or that you can see in the picture, and that's what causes it to fold in on itself to form a tertiary structure. And then your quaternary structure is going to be multiple tertiary structures all together to form one fully functioning protein. What is going to assist with protein folding is this thing called a chaperonin. So it assists in proper protein folding. So you're going to want to write that down. It assists in folding. And basically the polypeptide goes inside of it, it folds up, and then it leaves. And so this is just a protection. Kind of like if you were to go to like a store and try on clothes, you'd go into a dressing room, you'd try on the clothes, and then you'd walk out and look in the mirror. And so all I need you to know is that a chaperonin is a protein that actually helps with the folding of other proteins. And so the last thing that I'll say about proteins is that the structure and the function and the folding are very sensitive to chemical and physical conditions. And so all of those hydrogen bonds, ionic bonds, disulfide bridges, and the van der Waals interactions that make the protein fold into its very, very specific shape can become unfolded or denatured if there is a change in pH, a change in temperature, or if there are certain chemicals that it's exposed to. Sometimes a denatured or unfolded protein can renature and go back to normal. This is what happens whenever you have a fever. If your body gets really, really hot, some of your proteins probably denature a little bit, but then they can go back to normal so long as you stay as a human under 104 degrees Fahrenheit. Once you get above 104 degrees, that is when some denaturation can become permanent and you would end up with seizures, and that could actually result in death. And so it's really, really important that you get from this slide that the folding of proteins are very important to their function. If a protein unfolds at all, it can become completely ineffective, or it can't do its job anymore. And so you're going to have an assignment where you look at the denaturation of proteins and some of the things that can cause that to happen. And the little lab that you're going to watch has a woman who is working with albumin or egg white. And that is all I have about proteins. We will visit protein structure, function, and folding again. Like I said, this is a very important theme of this class. And that's actually all I have for you today. I will see you later.